Good afternoon. How is AI changing drug discovery, particularly in the field of flu? That's what we're going to be discussing today in this special roundtable for two companies that are collaborating in the area, Poolbeg Pharma, based in the UK, and CytoReason, based in Israel. My name is Stephen Adams of Optimum Strategic Communications, and I'm hosting today's discussion. Artificial intelligence is the hot topic in drug discovery right now. Many companies are turning to AI to improve their pipelines and to raise the efficiency of their research and development. How is artificial intelligence helping us discover useful drugs? What is it and how is it helping?
Yeah, if I may, I'll start off and just kind of give a bit of where we come from this at CytoReason. Basically, there's been an explosion that's been happening in biology over the past 20 plus years in terms of the ability to measure the complexity of the biology, especially in humans. So if you go back 20 plus years, people did experiments in biology, and it was one tube, one result. And it's today at a point where we take a single tube and we can measure, a single sample, and we can measure hundreds of thousands of genes, proteins, cells that are participating in particular disease or infection, as we have done here. That explosion of data obviously poses a great opportunity, but also poses a lot of problems of how do you actually discover within a field that is so rich that goes beyond the comprehension of humans and experts to grapple with everything. And that's where AI has been coming into drug development. Can we actually bring in that richness of data and bring in sophisticated analytical methodologies that start bringing in not just kind of the obvious things that we knew as kind of researchers, as scientists before, but the emergent information that comes out from this rich data that poses new opportunities that have yet to be explored, particularly in the cases of looking at the immune system, where the huge amount of variation in the human immune system has always been a barrier for development. And the new data that's now available and is actually part of this collaboration offers kind of an ability to explore human variation and how we manipulate the immune system to give better outcomes in disease.
I think if I could add as well, I like the idea of back when I was doing my my graduate and postgraduate work, it was that kind of one tube, one experiment and seeing where it goes. Very simple. I think appreciating, certainly from a Poolbeg standpoint, the data that we have from our human viral challenge studies, and with particular the CytoReason collaboration. This is 130 volunteers who were deliberately infected with influenza as part of the viral challenge study. It's challenging to analyze across the course of that infection, disease progression, resolution over the time frame. If you were an individual in a lab, working to understand the disease progression on one sample, it would be challenging. So when you have 130, that kind of adds exponentially to the complexity. So I think this is where CytoReason bring their knowledge and their expertise, and obviously their AI capabilities to the collaboration to help us understand what's going on under the hood, as it were, in a disease progression. And obviously when your numbers are larger, maybe you have greater confidence in the data that you see. And I think that's been a wonderful collaboration now since under a better understanding of the disease progression. But and again, the targets identified, we're very excited about these targets and progressing them into the true drug development process now. So I think that we're off to a very strong start.
Yeah, I just echo all of the comments that everyone's just made. It really is this boom of data that we've seen over the last 20 years. And I think pipelines are growing across the pharma industry, and there's so many more targets, more drugs being developed, that the difficult questions are, where do we prioritize our efforts initially? And the great thing about AI is it allows us to do things much faster, but also to have the insight of what Shai mentioned, so the human data. So we're not just looking at preclinical mice and then trying to figure out things at a later stage when we get to humans. AI can help us to model those late stage diseases. So we're making more informed decisions at an earlier point in time. And I think that's what pharma really appreciate from the models like CytoReason can offer, is that it brings that late stage insight into the early stage pipeline.
Okay, great. So AI is really helping us to crunch these numbers, these enormous data sets that we now have that we didn't have 10 or 20 years ago. Shai, you just mentioned one particular area was the immune system, and you said there's a huge amount of variation. That's a huge amount of variation from person to person, is it, in how people's immune systems deal with certain threats? Is that right?
Yeah, certainly, and I think the most recent pandemic really showed this. I like to think about the immune system as like a sixth sense. It basically monitors, it monitors the internal and external environment and responds to it. So ultimately, even if you have twins and monozygotic twins, just by living life, our immune system changes. And when we encounter an infection, people respond differently. That variation on one hand is a hindrance, it's an obstacle to understanding what's going on. On the other hand, once you start thinking about this from an AI perspective, where you're connecting the dots, there's a network of features that interact, you actually can leverage this variation to get more out, to understand more what's going on.
Jeremy, you touched on the central focus of this roundtable, the project that Poolbeg and CytoReason have going on at the moment to investigate flu. But if you can all, from both the Poolbeg side and the CytoReason, explain in simple terms what this project is. What are you doing and what do you hope to achieve?
Well, I think we start off with this unique data set. Yes, that I touched upon this. This is, healthy volunteers deliberately infected, they become sick and they recover. And we've got samples all the way along. These are, patient samples, so we can isolate, the gene expression and identify the gene expression profiles, protein expressions, etc., all the way through from, as I say, getting sick and resolution. So, to Shai's point, you can monitor, understand the immune system and what's going on as it progresses. I think what's unique is that we know the exact time of infection. So I think that's kind of a key, kind of when you're understanding your baselines, you take samples the day before infection. So you understand what this individual is like, what their immune system is like when they're fit and healthy, and then obviously progress looking at the immune system as it progresses through the infection and resolution. So having that kind of day -1, we call it, data is very important. So then you're able to kind of do it on an individual level, but then as we said, across the 130 volunteers, and we did compare it to external data. So it was a terrific amount of work. The data we have is very unique. Nobody else has that. And maybe I'll pass across to Liam to maybe go into the nuts and bolts of what was involved, certainly in the data set itself and the transfer.
Yeah, thanks, Jeremy. I guess one of the big challenges is what Shai mentioned earlier, that there's a lot of variation in disease, and we can get a lot of data out of samples nowadays. But one of the challenges to contextualize that data. So we all respond differently. We all have a different baseline. So when we're healthy, fit, happy, our bodies are very different. And when we get infected or we get a disease, our bodies change. But unless we understand that baseline, it's very difficult to contextualize disease. So having the human challenge data puts us in a really unique position where, because we have everybody's baseline data and a large amount of it, we're able to see in great granularity what's actually happening at the early stages of disease. So that, one, helps us to contextualize what's driving late stage severe disease. But secondly, it also allows us to target things that aren't being activated in that early stage of disease. Because when we're developing new drugs, what we want to do is to target parts of the immune system in the body that are involved in this pathogenic response, similar to our lead product, POLB001. We don't want to switch off people's immune systems. It's very important that they continue to fight the infection, but we want their body to actively and productively fight the infection without also damaging itself.
Indeed, the data set that Poolbeg generated was unique and allows very unique insights. And the place of AI in this game was, one, to analyze it, as Shai mentioned, and isolate the trends and understand what are the main themes across the variation in the patient population, or healthy volunteer population in this case. But it also allowed us to link this data and compare it to a set of data from severe patients that are hospitalized with the complications. And it's the combination between the severe patient data and observations of disease pathogenesis processes that were associated with severe disease outcome, and how do they reflect what happens in the normal so-called course of infection and those more healthy volunteers, that allows you to glean insight and try and tease out components of the immune response that might be more important in inflicting damage as compared to those that are needed to overcome the disease. It's the tools, the AI tools, that allows you to connect two completely separate data sets in two different clinical situations and have insight that might be informative for drug development.
This is a field in which people make very big, expensive mistakes. And what AI is here to do is actually make sure that they do less of that, and drugs become, more precise and with, and develop with less error and basically increase success rate. So that's where we see this as really kind of good.
Okay, so AI is giving a sort of a fast track to getting on the right roads to identify the right drugs, or drugs with a higher possibility of success. Is that a fair summary?
I think so. I think AI is the only way forward for what we call precision medicine. I don't think the present healthcare environment allows to get to this idea of the right drug for the right patient without AI being there contributing.
I think from our side, what I'd like to add to that is that quite often in research and development, it's never a straight line. It's a path that changes directions many times. And that's because generally we do it in a stepwise process where we get results, we interpret them, and then we decide what the next steps are. So quite often it looks in hindsight like it was very deliberate, the path that was taken, but it's not always the case. And sometimes that's because you're learning during the process. So having that late stage insight gives you the information that you kind of know where you're going, and that's a massive advantage. There's an old saying in pharmaceuticals that if you're going to fail, you should fail fast and fail early. And that's this whole part of increasing the chance of success, but also saving costs. And that is massive for us. So because we know where we're trying to go, that is the major advantage here.
Everyone, it seems, is doing AI analysis now in drug discovery world. How do you ensure that you're picking the right partner?
To speak on CytoReason's behalf, almost from Poolbeg's perspective, they were an intuitive fit for us. There was an extensive process that we went to find, the right partner for us. So we knew what data we had, and we knew where we were hoping to go. And a lot of AI providers provide slightly different services that are well adjusted for different people, or different companies. I think CytoReason were exceptionally well suited for Poolbeg. Shai himself and Yehuda have an extensive career in these areas. Extensive publications that marry well with the exact types of data that we were bringing. They have the preformed teams, and I think this is one of the great strengths, where they don't just have data scientists and AI teams, they also have immunologists, biologists, and bring all these interdisciplinary people together to work in harmony. And it's that multidisciplinary approach that we were able to work with, and bring all these different skills to get where we needed to go.
Thanks very much. Yehuda and Shai, can I ask, from your point of view, it takes two to tango. I should imagine you have lots of companies coming to you wanting to pick your brains and to apply your knowledge and your skills to their problems. I should imagine you don't say yes to everyone. What was it about Poolbeg that excited you?
So first of all, I want to echo and thank the comments just made. It's a pleasure to hear. And yes, certainly there's a lot of people knocking on our door, and it takes two to tango, and you want to get the right partner for this. And so Poolbeg, both, I mean, I cannot stress how unique what that data that Poolbeg brought to bear into the collaboration is, and how the strong belief, and I think this is Jeremy expresses his enthusiasm, is within CytoReason that with the right data, you can actually work magic and really forward drug development. You can, we talked before about de-risking and making less mistakes. It takes the right data to do this. No AI, with nothing in the air, you're only guessing. If you have the right data set, you can do a huge amount. So that, and then the other side is the people, the scientists, the relationships. Of course, But that combination, the whole idea of performing challenge studies in humans to study the diversity, to start the time course, to see how disease begins and then resolves, is how we believe it needs to be done. As Leah mentioned before, Yehuda and I have done this and published on this in academic settings, but you never see the type of data that Poolbeg brought in, and suddenly you see this, and this could be a success for us. This was important. Poolbeg came out 2 years ago, and it was we saw this as, yes, this is something that we can see developing, expanding, and succeeding, and that's how we make the choices. Ultimately, it's people and the quality of science and so forth that comes in.
So having this human challenge trial data is rare, is it, Shai?
Extremely rare. Extremely rare. I mean, Jeremy can probably comment more than I, but the number of such data sets out there are very limited, and none with the richness that Poolbeg has put into generating this with HV.
What you've achieved so far in your collaboration, what are the outputs that you've got, and where do you hope to take those outputs?
So we are now coming to the end of the collaboration. We have identified a number of targets that we're very excited about, some of which are completely novel for the treatment of influenza. So the next steps are really to get these into the lab, to validate them and move them forward. So they come with data packages that will help us design that process. So really what Poolbeg are trying to do is to move that forwardly as rapidly as possible for these targets.
And can you explain in simple terms what these targets are? Do they act on the virus itself? Are they antivirals? Are they something else?
So our approach has been different to the current marketed products. So the existing marketed products for influenza are largely antivirals, which means that they in some way disrupt the life cycle of the virus molecule in that it can no longer replicate. The Poolbeg approach has been to identify host-based modulators, quite often immunomodulation, so targeting the immune system. And the idea here is not necessarily to primarily combat the virus life cycle, but to instead look at how the human is responding to disease, to ensure that that response is an effective response, because people can have immune responses, and as we get older, our immune system tends to be a little bit more challenged. It tends to react a little bit less, and then when it does react, quite often it can go overboard. So what we're trying to identify is to modulate the host, the human, so that they have an effective immune response from the virus. The hope here is that if you have an effective immune response, you clear the virus quicker, and you also experience significantly less disease.
This is where the AI methodologies kick in, because what you really learn are processes that cause tissue damage, and you try to isolate which part of the immune response is inflicting damage to the tissue, which are destructive, and this can be extrapolated to other diseases as well. This is the way that you connect this very unique data set to clinical settings in the context of flu and other viruses or other diseases that have mimicking damage in the tissue.
Do you have other particular diseases in mind, or is it too early to say that sort of thing?
The most recent difficult and deadly was COVID, where you have an immune response that, especially with the first variants of the virus, that were very destructive to the lungs, and that were caused by the immune response, not by the virus. And indeed, patients were empirically treated by immunomodulators, those who were available. Now imagine how better the situation could be if you come up with immunomodulators that are directed at processes that are known to cause this particular lung tissue damage. It can potentially be less harmful from the point of view of side effects, and more effective from the point of view of efficacy. So this is just a very clear and immediate example.
the virus itself, it drifts. There's kind of antigenic drift, and it kind of changes. And similarly with the influenza, there's different strains emerge every year. But I think we're coming at this again from this universal immune response. So regardless, certainly in the influenza side, regardless of the strain that emerges, every flu season, we hope to have a drug that'll be able to tackle that particular virus. And I think Yehuda kind of alluded to the fact that maybe it goes beyond just influenza. We also have a program around looking at RSV in our collaboration, let's say from with our agreement with hVivo. They've got other challenge studies that are going on where we've could have access to that data as well. So we can go beyond and just influenza or RSV or indeed COVID. So coming up with a universal drug, a universal response. I mean, it's almost like a holy grail in a sense, but that's kind of something that AI could point us in the direction of.
So just to reiterate there, Jeremy, you're hopeful, are you, that your candidate could help patients with all sorts of strains of flu?
That's the plan, yes. I mean, we start with POB-001, which is P38 MAP kinase inhibitor, and we know that in that transition from mild to moderate influenza, that P38 is increased, and P38 is an immune modulator, so that's ultimately driving the cytokine storm that causes damage. So we're looking for other parallels there. I think what was interesting as part of the collaboration, we were happy to see that P38 MAP kinase did come out, in the CytoReason analysis to be a target of interest, and obviously there was others that we went through as well, but it is the driver of that disease, of that cytokine storm. So if you block that, then you're helping to calm the immune system down, so you're not getting that tissue, lung damage, vascular damage, etc.
Is this lead candidate? Is this something that you could potentially try in COVID or not?
Well, I think that the answer to that will necessitate looking closely into the COVID cohort to understand what's going on there, not to make an empiric assumption, but to look at the data and decide based on the data. And I think that definitely, for instance, pathways or algorithms or approaches that were trained on influenza can be used to look at other data sets, methodologies or logics that were fostered together in collaboration with the Poolbeg scientific team. These were very productive discussions because they brought two different approaches together. Can be used to look at COVID. So I definitely think that this shortens the path to examine the relevance. And by the way, this is something that is typical of the type of work that we do in CytoReason, that we could think of a problem for the first time for a given amount of time, and then do the second round in a much shorter way, more efficient and direct, because we've thought about the problem and we don't throw it away. It sits in the algorithm, waiting for the next opportunity.
Okay, and just drawing it out wider, cytokine storm isn't just a side effect of infectious diseases such as flu and COVID, is it? As I understand it, you can have cytokine storms when other things are happening. Could you apply the knowledge that you're gleaning at the moment to completely different disease areas?
I think we're all speaking like scientists here, where we don't, we want to see the data first, but it's like the potential for sure is there. But obviously we want to kind of understand the experimentation behind it. Let's see the data. But again, we're looking here. I think if you think about back to the immune system, the innate immune response, the first reaction is where the immune system doesn't realize that it recognizes there's an infection, but doesn't realize what the pathogen is. So there's this non-specific reaction to the presence. So we could be targeting something there that kind of that blocks something universally. But obviously the next stage then of the immune system is getting a bit more targeted, to the actual virus in particular. And we always use the spike protein as an example for the vaccine generation during COVID. But targeting that initial innate immune response, we could find something quite exciting that has a role to play across several diseases. But as I say, as scientists, we want to see the data first before we fully back it.
I just want to point out that when you identify a pattern using these computational methodologies, then it's much easier to screen across different pathogenic states. And sometimes you come up with surprising answers you didn't anticipate to begin with, because the data informs you that there might be something there that would be worthwhile to look into. Just to give you an example, an unrelated field, there are drugs that are used across diseases in the GI rheumatology field, and these, you wouldn't anticipate that just by thinking about it in an abstract form. But today, when we look at it, we could see patterns. And what we've done here together, and this collaboration could serve as a basis for screening other things that will necessitate deeper look into the data to take decisions.
Yeah, that's a good point.
So more generally, if we're thinking about AI, is it helping us to think more laterally about drugs and drug targets, and how you might start in one disease area and think, well, we could use either this drug or this approach in a completely different disease area?
Very much so. This is actually at the heart of the CytoReason technology. We developed the core of the technology is what we call computational disease models. So we model every disease, bringing in all the data that we can, and kind of modeling all the biological entities in it and how they change over time. What are the cell states, and the variation between individuals, and how this affects treatments. And we do so for many, many different diseases. And so that gives us an opportunity, once you make a discovery in one, and this is kind of what Yehuda was alluding to, you can now take that discovery and with low cost, no new experiment, just first try it out in the computer, test all the different other diseases, test subpopulations within the other diseases, and ultimately, the way we kind of envision this, I think how this industry is moving, and I mentioned this before when I said precision medicine. If you look at this, the drug development industry today, much of it is about getting the blockbuster, that drug that works for enough of the population that the company becomes rich from it. And of course, it's hard to keep coming up with these, and ultimately, if you look, there's actually not that many, right? But with AI, one could start imagining what you could call a string of pearls type approach, where you start building a strategy for drug development that starts in the computer and says, okay, I think I can target this subpopulation in this disease, and that subpopulation in this disease, and that subpopulation, and I can build a commercial offering that would involve this, and then I start the clinical trials, right? And the first one obviously would go with the place where you makes more sense, as Poolbeg has done here with flu. But as Yehuda alluded to, you can now start doing on the computer a lot of that. You have just generated high value data that you can try to fit as a pattern in all these other computational models. And so that, I think, is part of where we're going as an industry, is more of that going on initially.
Yeah. I think it's a case of like reducing risk we touched upon earlier on, but the time saving, and again, we link back here the story at Poolbeg with the lead program POB-001 that was identified as I mentioned earlier, a key driver of severe influenza, but it took them many, many years of laborious, and I mean laborious, the laboratory exercises to kind of tease out the role of P38 MAP kinase in severe influenza. But our collaboration with CytoReason, I think it's 15 months or so, we've taken a larger data set. we've interrogated this large data set. So we've coming up with these, novel targets that, as Liam mentioned, there were, I think we all appreciate that as described, the low-hanging fruit have been removed for years. So now we're looking at kind of more complex disease, more, but again, it's more, an understanding of the complex disease. But the targets coming out are equally as valid, and they're kind of hidden behind. And I think the short space of time has just been remarkable as well. Just the pace of the advancement of science and understanding of disease, I think, in this short space of time. And then what we believe to be a greater probability of success with the targets identified. I think it's a win-win. A shorter time frame and a greater probability of success. So it's a win-win from an AI standpoint.
We've talked about AI mainly in terms of it helping with drug discovery and helping refine the potential pipeline. does it also have a role to play or potential role to play further down in terms of validating those early findings and in terms of the clinical trials which are needed to get approval?
There was an amazing decision made by the FDA this year, and that is that you could file for a drug without doing animal testing. Right now, if you take this idea, now nobody's done it yet. That decision is going to take time until it happens. But it shows you a change in mindset that if you go back a few years ago, no one would have said that this could happen. Right? That the FDA is going to allow you to develop a drug without animal testing. And I think that kind of takes us to where this field is going. Right? Yes, it's going to go more and more into regulatory decisions. It's going to save money. It's going to accelerate decision making. We have done so even within our context of some of our collaborations during COVID. Pull some of the information in, bring something to the regulator much faster for initial approval. So this is a shifting landscape that I think in many ways has been slow to move, but ultimately is going to be very different than how we see it today, and it's going to play a significant role in this.
Okay. Thinking, broadening things out slightly and thinking about pandemics, both past, one might almost say present and future. The capabilities that we now have in AI in mid 2023 are beyond the capabilities that we had 3.5 years ago. We had that DeLorean time machine. And if we could jump in that car and go back 3.5 years with the capabilities in AI that we have now, do you think those could have had a material impact on how the pandemic played out? If so, how?
I'll take a first stab. I'm sure everybody has something to say this one. I think yes, they would have made a change, but in my opinion, only marginal. And the reason is because simultaneously to a development of technologies, what also developed is the ability to generate data and quickly, right? And we saw this with the type of data that Poolbeg has brought into our collaboration, right? When COVID started, data was sparse. Within this context, data started coming out more, right? And now in a new setting, in a new pandemic, we can generate data much faster than we could before. I think that piece goes hand in hand with AI. Data and AI, that is a wedding, right? And that's, I think, a part of why this collaboration is successful. So it's not just about the AI, it's about the data. Now, if the AI that we have today was in place then, and the data was coming in quickly, we could have churned out much faster some of the decisions that
I guess it would have been very useful to go back to the very early stages of 2020 and convince people to do human challenge trials right then.
Yes.
And applied AI tools to those human challenge trials.
Yeah,
that would have been nice.
Right, but that's a wonderful example. You needed the challenge studies with the AI, right?
Yeah.
Okay.
No, that's a good point. Yeah.
Thinking more practically about the future, COVID surely is not going to be the last pandemic, not going to be, but by a long shot. Where do you all hope that the science and the technology will take us in terms of being able to deal with the next pandemic?
Yeah. I'll start here. I'm sure that everyone has an opinion, but I think you're right. It's with regard to the next pandemic. It's not a case of if, but when. I think there's concerns that indeed influenza is something of concern. Again, I mean, there's been incidences of, say, bird flu, and it's kind of jumping from species to species. But I think what we hope, if I be so bold, from this collaboration, is that we will have drugs ready, for the next pandemic. That we've identified through this collaboration that is stockpiled somewhere. We've been through the process of drug approvals, etc., and then a stockpile, so that if there is that emergence of the next pandemic, influenza or otherwise, there are drugs that have been, without the, let's just say the shotgun approach that happened during COVID, where they just tried any immune modulators, for example. We have something a bit more targeted, that we'd have a drug that would be able to roll out, mass markets, get it into the hands of those that need it very, very quickly. So again, we're trying to get ahead of the curve when compared to what happened in the COVID pandemic, where they were still figuring out what exactly this virus was for in the early days of it. So we hope to be better prepared. It's all about pandemic preparedness now, and there's a lot of governments are buying into the fact that they need to be better prepared the next time around. And once it's identified what that strain is, they'll get into action and start saving lives as soon as possible.
Maybe just from an AI perspective, I totally echo what Jeremy said. This is where the field is going, and people want to be prepared ahead. Also comes in from the computation, from monitoring monitoring for pandemics, understanding what's happening in the infrastructure is being put in, or people are thinking about this, and so COVID has totally changed how we view, I think, drug development going forward.
Okay. And finally, thinking much more generally, not just about pandemics, not just about flu, but about drug discovery at large. How is it going to be conducted in the medium and the longer term? Now, this is really future gazy stuff. But if we're talking the timescale of say 10 to 50 years, how things are going to be different?
I'll take a first stab. I think about this problem often. I think it's actually a good driver in this field where so much technology and capability are coming out, data generation and AI. We need to be thinking ahead. And the answer, while I don't know it, and I don't think anybody knows, there are some principles that we get. And less mice. I said, Yehuda said this before, and the FDA just gave us a very clear signal. More automation in experimental work, so the biologists are actually shifting in some ways what they're doing. They become, they are data scientists. The data comes to them, and they think about biology in this sense. So they're a new flavor. They're data users, if you like. The data science is the new pipette, right? So more computers, less mice, and more automation in experiments. And of course, and I think just kind of Jeremy was saying, earlier preparedness in this. So kind of picking up subpopulations, picking up that would be kind of relevant for particular drugs, picking up preventive medicine. I'm excited by this future, and I think the way to get there is to start now, and that's, I think, what this collaboration has been about. So.
Yeah. I agree with Shai. I think that there's a saying in Hebrew that prophecy was given to the fools. So I'm sort of being a bit cautious here.
Super. Well, it's been a fascinating discussion. Thank you all very much for your time and your thoughts. Thank you to Jeremy Skillington and Liam Tremble of Poolbeg Pharma, and Shai, Shen-Orr, and Yehuda Chowers of CytoReason. Thanks very much.
Thank you very much.
You.
Thanks.